would say like the two main things that limit our lab findings from getting into people are, number one, a lot of things work in mice that just don't work in people for whatever reason. Like there are a number of reasons, but you really need to have like a slam dunk in a mouse experiment. You need to cure the tumor and it never comes back. And that mouse is like running around and happy and healthy and perfect. Because if you can't do that, you're not going to have good results in a person. If you have like just okay results in mice, it's going to fail miserably in people. People are harder. They're complicated. And like genetically, we're so different and we have different mutations in our tumors, and it's just harder in people. So things just fail in people that don't fail in mice. And then the other thing is it's stupid expensive to do this research. Like to make a batch of GMP grade antibody, like the antibodies that we would give to people have to be made in a special clean lab. And everything is really regulated and for good reason. Like the stuff we make in the lab, you don't want us to inject that in people. It's like dirty and sketchy. So before you do a clinical trial, you have to do what's called GMP manufacture. It's like good manufacturing practices. And you contract that out to an organization that manufactures drugs in that way. But the cost to get enough antibody to do a phase 1 clinical trial is probably like $7 million.
Wow.
So we can't just come up with an idea in the lab and immediately test it or test like 10 different things. Like we really need to prioritize what we think has the best chance of working. And then we have to get money. So we can get grant funding or we can get internal funding from MSK. But it's almost always a combination of those things. And we can also get investor funding. We can license the technology. So if we invent something at MSK and we have a patent on it, we can license that to a biotech company that has a lot more money and that can pay for that research. But it's like a whole other part of science that I never had any idea about until I came to MSK and I came to this lab that's like very focused on drug development. So it's been a real learning experience to figure out how do you get the money to turn this idea into a trial. And for DSRCT, it is so especially hard because it's so rare, right? You are not going to get a pharmaceutical company to give you $7 million to make a drug that is only specific to a couple hundred patients a year. No matter how nicely you ask them or what a great story you have, it's not going to happen. So the way that we've gotten around that is all of the antibody targets that we're looking at are not just expressed on DSRCT. They are expressed on DSRCT, but they're also expressed on breast cancer or lung cancer or colon cancer. And then all of a sudden, these companies are much more interested because they can do a nice thing and help you cure the rare cancer. And they can also potentially make a lot of money, which is fine. I'm happy for them to make money if we can also get something into trials for these patients who have not a lot of other options.
No, I think those are all great points. And I think it's, one of the things that I try in coming up with ideas for how our community can better advocate for progress. One of the points I try to make to people is that because DSRCT is so rare, the likelihood of a technology that treats DSRCT getting funded is small. But if you consider that a slam dunk for DSRCT is going to cure many other people, it'll be translatable to so many other rare cancers and sarcomas, and even outside of sarcoma, that the investment there is a return on investment, because even though we're looking at it as a couple hundred patients will benefit, that's not really the case, because what we learn in DSRCT can impact so many other types of cancer communities.